Everybody to our first webinar in 2021. I hope you all had a good start into the new year. My name is Tobias Fischborn and I'm the R&D Manager for Lallemand Brewing. I will be your host for today's deep dive into yeast nutrition. In brewing, we often believe that the malt wort provides sufficient nutrients to the yeast. However, this is not always the case, as certain nutrients like minerals, for instance, be present in the wort, but not available to the yeast itself. In high gravity brewing or when the malt quality is poor, the demand for extra nutrient addition is even higher. And then there is mead, cider, and the increasingly popular hard seltzers. These are very challenging fermentation media with very little nutrient content. So a good nutrient management, how much of what and when, is essential for these kind of fermentations. I'm very happy that we have two experts on yeast nutrition with us today that will share their knowledge. Our first presenter is Anne Julien-Ortiz. After a master's degree in food engineering and at the Institute of Sciences in Montpellier and specialization in microbiological engineering, Anne joined Lallemand Oenology department in 1999. Her first work within INRA Montpellier in France at the Department of Sciences for Oenology focused on yeast nutrition under oenological conditions and its influence on yeast physiology. Today, she leads research and development projects at an international level on yeast and their nutrients and initiates research collaborations with research institutes worldwide dealing with the metabolism of wine yeast. After several studies on the characterization of wine yeast and the biodiversity of yeast, which led to the selection of yeast on world's terroirs, most projects now focus on the improvement of specific properties of wine yeast and the development of yeast nutrients to optimize the organoleptic quality of wines. Anne will present today the different phases of nitrogen in alcoholic fermentation, impact on yeast metabolism. Our second speaker is Professor Graeme Walker from Abertay University, Dundee in Scotland. Graeme graduated with a BSc in Brewing and Biochemistry in 1975 and completed his PhD in Yeast Physiology in 1978, both from Heriot-Watt University in Edinburgh, Scotland. He did his postdoctoral fellowship at Carlsberg Breweries in Copenhagen, and in 2004 he obtained a Doctor of Science from Abertay University. He's currently the professor of Zymology at the University of Abertay Dundee in Scotland and leads a yeast research group there. Graeme is also the scientific director of the Ethanol Technology Institute, acts as a guest lecturer at the Siebel Institute, World Brewing Academy in Chicago, and consults with the brewing, distilling, and the fuel alcohol industry. He has also published over 200 articles and authored several textbooks, including Yeast Physiology and Biotechnology, Bioethanol, and the Alcohol textbook. Graeme will talk about brewing yeast nutrition. So let's start our deep dive into yeast nutrition. I would suggest that you write your questions in the Ask Questions section and that we will answer them at the end of the second presentation. So Anne, if you want to share your screen and start.
Is it okay?
Yeah.
Okay.
Perfect. So I will go quiet.
Okay, so thank you, Toby, for the nice introduction and for giving me the opportunity to share with you, all the worldwide brewers community, a new insight into the different phases of nitrogen in fermentation and its impact on yeast metabolism. The objective of the presentation is to come back on some basics on the yeast nutritional and nitrogen needs in alcoholic fermentation, and to illustrate as well the importance of nitrogen sources on the yeast fermentative performance, but also on the fermentative and varietal aromas. The last part of the presentation will introduce new knowledge on nitrogen interactions with other key compounds and what is the impact of such interactions on yeast viability and flavors. So during alcoholic fermentation process, yeast ferments sugar into alcohol and carbon dioxide, biomass, and other byproducts which participate to the organoleptic properties of fermented beverages like beer or wine. These compounds are organic acids, but as well esters and higher alcohol. To achieve successfully these processes, yeast requires specific compounds, normally naturally present in its environment, called nutrients. These nutrients are mainly the nitrogen, vitamins, minerals, but also lipids, which are compounds of sterols and unsaturated fatty acids. So all these nutrients are necessary for the yeast to grow and to ferment all the carbohydrates present in the medium, could be glucose, fructose, but as well maltose or maltotriose. So the topic of this presentation is to focus on a little bit more on nitrogen. And when we speak about nitrogen in alcoholic fermentation, we mainly speak about the nitrogen part, which is actually assimilated by the yeast. And this yeast assimilable nitrogen, that I will call YAN for the rest of the presentation, is composed of ammonium, free amino nitrogen, minus the proline that the yeast cannot consume, and small peptides. So the YAN, all these nitrogen sources, is a key nutrient because it determines the yeast growth, but also the fermentation rate. The level of this yeast assimilable nitrogen can be really variable in wort and can be often deficient. It impacts strongly on the composition of the beer or wine and the flavor of the products. And we will see that yeast vary widely in their nitrogen requirements. If we look closely on the fermentation process, we can divide the life of the yeast in three very important phases, three different phases. For each of them, specific nutrients are essential. For instance, the first one, which is the lag phase. During this phase, vitamins and minerals are the key compounds because they act as cofactors of key enzymatic reactions, and they allow the yeast to optimize its capacity of adaptation to the medium, which is usually a hostile environment, and to take place in the medium. The second phase is the exponential or the growth phase. During this step, nitrogen here is really the key compounds because it is really essential for the protein synthesis and, of course, the sugar transport into the cell. In this way, it allows the yeast to optimize, to maximize its multiplication rate, to reach a sufficient fermentation rate, reaching as well a maximum of biomass production. All the nitrogen is usually consumed during this step. And finally, the third part of the fermentation that we called the stationary phase because there is no more multiplication and growth. All the yeast have already reached the maximum of population. During this phase, the nitrogen has been consumed, but there is still a lot of sugar to be uptaken, with ethanol becoming higher and higher. So to secure membrane integrity and maintain the viability, it's really important during this time that the yeast accumulated sufficient sterols and lipids, which will be really useful for the yeast to maintain at the highest level its viability. Coming back to yeast nitrogen requirements, they are a high variability depending on the yeast strains. And if we have a look on this graph here, which illustrates the milligram of YAN of nitrogen sources, assimilable sources, necessary to consume 1 gram of sugar, we could see that some strain needs 4 times more nitrogen than others to ferment a deficient nitrogen medium at the same fermentation rate. So there is a huge variability depending on yeast strains regarding these nitrogen requirements. In the case of nitrogen deficiency situation, we can see that if we add nitrogen at the beginning of the fermentation or even during the alcoholic fermentation, it really helps the yeast fermentative capacity, allowing the fermentation duration to be drastically decreased. So we can see on this graph a fermentation control in black with a black curve, which represents the fermentation rate evolution in function of the time of the evolution of the fermentation. And we compared for the same yeast the supplementation of nitrogen at the beginning of the fermentation, and we can see that it impacts drastically on the fermentation rate, decreasing by 5 days the fermentation duration, corresponding more or less to a reduction of 30% of the fermentation. But the yeast nutrition management in fermented beverages evolved quite a lot this last 20, 30 years. And actually, what's true is that in the 90s, the first objective was really to limit the risk of stuck or sluggish fermentation. And people using nitrogen to secure the fermentation focused really on these compounds because it was defined to be the most important for the biomass and to have a direct impact on the fermentation rate, improving the yeast fermentative capacity. Sorry. Up. Then, 10 years ago, thanks to new studies, it has been shown that all nitrogen sources were not really similar. The nitrogen quality, meaning organic or inorganic, became the new focus of investigations. And free amino acids and peptides have been shown to be a better nitrogen source compared to inorganic source like ammonium, for instance. And we will see that organic nitrogen has been described to be more efficient on fermentation for the same yeast assimilable nitrogen level added. And we are going to see how it impacts positively on the fermentative and varietal aromas. Okay, sorry. So in this work, which has been realized with a famous wine institute from Australia, it has been demonstrated that with 2 times less nitrogen added in the medium, here it was a Chardonnay juice, from organic sources, the fermentation was faster than the fermentation with inorganic nitrogen addition. And we compared ammonium addition with a level of YAN 2 times higher than the fermentation only with organic nutrition. And we could see that the fermentation was shorter with the modality with the organic nutrition added, even with the lowest total yeast assimilable nitrogen addition, meaning that organic nitrogen is significantly more efficient than inorganic nitrogen to complete the fermentation. At least in this example, we can see 2 times more efficient. Sorry, I have some difficulties to go through. Okay. Same kind of observation, again with the same institutes, On the aroma profile, with an enhancement of the fruity and the floral notes, with the addition of free amino acids, compared with the addition of inorganic nitrogen sources, which is responsible in this experimentation of an increase of acetic acid notes and nail lacquer of flavor. As we can see here. So nitrogen, different nitrogen sources have really different effects upon yeast response, including yeast capacity to synthesize aroma compounds and aroma differences. We can see aroma differences at the same yeast assimilable nitrogen, for the same yeast assimilable nitrogen level for ammonium versus amino acids. So now the question is how to explain that nitrogen has such an impact on the synthesis of aromas by fermenting yeast. So the metabolic pathways are well known today, but they are very interconnected and interdependent. And the regulation of such pathway is really complex. And when we speak about fermentative aromas during alcoholic fermentation, that means that these aromatic compounds are produced thanks to the yeast metabolism during the fermentation from specific precursors, sugars, and free amino nitrogen. And these specific compounds and fermentative aromas are called the ethyl esters, the higher alcohol, and the acetate esters. So if we do a deep dive into ester biosynthesis, we can see that nitrogen is involved in many pathways. And one of them is today well described. It's the Ehrlich pathway. And this Ehrlich pathway leads to higher alcohols and ester synthesis through free amino acids precursors. We can see that the amino acids are transformed through transamination and decarboxylation reaction into keto acids, aldehyde, and higher alcohol. And thanks to specific genes, today well identified, ATF1 and ATF2, the higher alcohols can be synthesized in esters acetate. But some specific amino acids are also direct precursors of ester acetates through other pathways. This is, for instance, the case for the phenylalanine, which through the pentose phosphate pathways can give very interesting aroma compounds like the 2-phenylethanol and the 2-phenylethyl acetate, which are responsible of floral and rose notes. Others, like the valine or the leucine, are directly issued or the direct precursors of cetoacid issued from the central carbon metabolism of the yeast. So we can see the various pathways where amino acids are involved to synthesize and to participate to the aroma profile of the fermented beverages. Here, I will present some results obtained through a Lallemand PhD realized 6 years ago in collaboration with the INRA of Montpellier in south of France. And we have investigated the nitrogen impact on the ester biosynthesis kinetic profile. So for such purpose, online monitoring of fermentative aromas was used. And we could see that, for instance, the esters are produced from the beginning of the alcoholic fermentation. If we have a look on the graph here, representing the CO2, the progress of the fermentation in function of the sugar consumed, we can see an example of the ethyl octanoate production during the fermentation. And there is really a production from the very beginning of the fermentation till the end. What we can notice is that the yield of the second phases is systematically higher than the first phases. And we could observe as well, following the production of several aromatic compounds in function of the level of the initial nitrogen, that the esters production by the yeast during this fermentation increases with the nitrogen level increase. So the highest nitrogen at the beginning, the higher ester production. So to go further, and with the target to identify the optimum fermentation conditions to produce specific aroma compounds, we have designed an experimental plan called the box Benken to evaluate the impact of environmental factors such as nitrogen and lipids on the capacity of the yeast to synthesize aroma. And we could define for yeast strain that, for instance, the optimal conditions to maximize the phenylethanol synthesis responsible of the floral notes was a medium range of nitrogen with a medium content of lipid. It's what we can see on the first graph with this nice profile with the red and the pink color, representing the phenylethanol production in function of the level of lipids and nitrogen. And we can see that the optimum is reached for a medium range of nitrogen and medium range of lipids. And we have applied this for more than 30 different aroma compounds. And for instance, for the isoamyl acetate, we can see that there is a strong impact of the nitrogen level, whatever the lipids level is. And for ethyl butanoate, it's another kind of profile. So this tool is really powerful and interesting to define the best conditions of use of a specific yeast in function of environmental factors such as the nutrients. And what's interesting is that I give you here just an example of what we could obtain for several yeasts. We have then investigated such kind of profile for different yeast strains, and we can see that, for instance, for one compound, the phenyl ethanol, all the profiles are quite different depending on the strains. More recently, small peptides have been deeply studied, and more yeast peptide transporters have been identified in Saccharomyces cerevisiae. So we can see some of them represented here. And it seems that peptides uptake affects, first of all, the pathways of carbon and nitrogen metabolism, leading to an increased de novo synthesis and availability of cetoacids, which are the precursors of the aroma compounds, and increasing the synthesis of some esters. So this has been published. And so we wanted to evaluate this observation. And thanks to our colleagues from Tallinn Competence Center in Estonia, we have validated recently the strong impact of such peptides as nitrogen sources on the aromas during fermentation. And it's what is illustrated with these three histograms, which show a very strong effect on ester compounds of peptides versus control on a deficient nitrogen medium and versus a fermentation with ammonium as single nitrogen source. And we can see that even if in the modality with ammonium has a single nitrogen sources, this level was almost 2 times higher than the total nitrogen added with the peptides form. We can see the significant positive impact on the acetate esters production with the organic nitrogen source, such as peptides. So it's really significant with these three specific aromas responsible of fruity or fruity notes in fermented beverages. Now, what about nitrogen and another kind of aromas that we call varietal aromas? So the varietal aromas are coming from odorless precursors from the medium that we can find in wort, in must, etc. And what we smell when we drink beverages are the volatile thiols, which are the odorant compounds mainly identified today like specific molecules of 4-MMP and 3-MH, 4-mercaptomethylpentanone and 3-mercaptohexanol. These compounds are released during the fermentation, and we will see that the yeast has really a key role in the revelation of such aromas. What is the mechanism behind? There is two steps. The first one is the uptake into the yeast of the precursors present in the malt or in the must. They are well known. They have been widely described in the literature. And these compounds are cysteinylated and glutathioneolated precursors that we can find in the medium. And these precursors are uptake thanks to specific transporter of the yeast cell membrane into the cell. And then thanks to the cysteine sulfonylurease of the yeast, of most of the yeast, with a different level of expression, of course, depending on the yeast genetic background, but most of the yeast get this beta-lyase activity. These precursors, non-odorant, odorless, sorry, will be released into compounds called the volatile thiols, like 4-MMP and 3-MH, which are responsible of blackcurrant notes or grapefruit or box tree, usually. But the nitrogen source or quality impact as well on the precursor uptake and on the capacity of the yeast to integrate these precursors. And this phenomenon has been well described in a PhD, which demonstrated that the release of volatile thiol finally is governed by what we call the nitrogen catabolic repression. What does it mean? It means that in the presence of ammonium in excess, the yeast will firstly uptake this ammonium, and this leads to a retroinhibition of some very important proteins from the cell membrane, the general amino acid permease, which allow the uptake of the free amino acids from the medium, but which are the same proteins responsible of the uptake of the volatile precursors, the cysteinylated or glutathioneolated that I described in the previous slides. So this they demonstrated effectively in specific medium that with a significant ammonium addition at the beginning of the fermentation, we observed a decrease in comparison with the control without any ammonium addition, a decrease of the interesting aroma, the volatile thiols production level. So to summarize this part, we have seen that nitrogen has several phases. This nutrient is essential for yeast growth, definitely. We have seen its high efficiency, especially of organic nitrogen source on the fermentation rate, the yeast activity. We have seen as well that nitrogen has a strong contribution on yeast metabolism to produce aromas, esters, but also volatile thiols. But it's important to know that nitrogen is important, but not on its own. And there is strong interactions with lipids and vitamins mainly, which impact on yeast variability, aromas, or flavors. And it's what we are going to see in the last part of the presentation, only 2-3 slides more. 2 years ago, nitrogen interactions with other key nutrients like vitamins, but also lipids, were studied, and especially what is the impact of such interaction on the yeast viability. So the first graph here, sorry. I have an issue. So, yeah, the first graph here, represents the viability evolution in function of the CO2 production in two kinds of modality. I mean, in this fermentation, we have low pantothenate level, but in one fermentation, the level of yeast assimilable nitrogen is low, roughly around 100 milligram per liter. In the other, the level of initial yeast assimilable nitrogen is high, over 300 milligram per liter. And we can see that in case of unbalance between pantothenate level and nitrogen, we observed a drastic, strong mortality at the very beginning of the fermentation, leading to a stuck fermentation and more than 90% of mortality. So we can see here that pantothenate starvation triggers cell death in case of unbalanced nutritional conditions. Another team from the Washington State University have demonstrated that this pantothenate deficiency can lead to a high H2S production. And this observation was even more important in case of high nitrogen concentration at the beginning of the fermentation. In this histogram, we can see for two kinds of conditions in terms of nitrogen level, 60 or 250 milligram per liter, with three different levels of pantothenic acid, that as soon as we have a pantothenate deficiency, the H2S production by the yeast is significant, and this increase even more in case of unbalance between nitrogen and pantothenate level. And we have seen the same kind of impact on the yeast viability, studying the nitrogen and the lipid interaction. And this has been done by a team of the INRAE in Montpellier, where we can see here, if we follow the viability in function of the CO2 released, meaning in function of the fermentation progress, that in case of high lipids content in the wort or in the must, whatever the nitrogen level is, we maintain the viability till the end of the fermentation. As soon as we are in lipids deficiency, with the three different levels of nitrogen, from low, medium, or high yeast assimilable nitrogen, we observed a mortality. So we can say that really lipids deficiency can lead to a high mortality, whatever the nitrogen level is, and that this mortality rate is modulated by the nitrogen concentration. The highest nitrogen with low lipids, the highest mortality. So to conclude, on the yeast nutrition management in alcoholic fermentation, we can see that the evolution over the last years have been very important. These last 5 years, we have observed new understanding, new findings, and we are sure today that yeast really need a well-balanced nutrition, that nitrogen source management is really important. Yes, it's key, but not only. Vitamins are key compounds, minerals, and Graeme will detail this part in the next presentation, but as well sterols are key compounds. And the goal to maintain a well balance between all these nutrients is really to limit the risk of nutritional imbalances, which can be, as we have seen, responsible of a loss of viability and yeast activity, but as well, which could develop of flavors like sulfur compounds production. So I hope it was clear enough. Don't hesitate if you have any question. Thank you very much. Thanks for your attention.
Well, thank you very much, Anne. That was a fantastic presentation, and I see we have already some questions, but as I said, we're going to answer them after Graeme's presentation. So I will try to share now my screen because we have some problems with Graeme getting his presentation up. So let's see if that works. Good. I guess we can all see. So Graeme, up to you, and just let me know when I should.
Okay.
Switch the slides.
Thank you indeed, Tobias, and thank you also to the Lallemand Brewing team to invite me to give this presentation. Incidentally, it's now afternoon time in Scotland and the pubs are closed, so I hope you'll forgive me celebrating Robbie Burns from yesterday, so Flangevar. Anyway, more seriously, next slide, Tobias, thank you. So yes, what I'd like to cover is aspects of yeast nutrition which are influencing the fermentative performance of yeast, but also nutrition that impacts on the flavor characteristics and the aroma characteristics of beer. And Anne has very beautifully demonstrated some aspects of that relating to nitrogen nutrition. I will cover a little bit more on what I've designated as minor nutrients, minerals, vitamins, and oxygen. But don't think the minor means minor importance. These are very important nutrients and are very often overlooked by brewers in terms of what is available in the work. Next. So yes, this is a very simple slide just to get going, and it's really considering the work that we make in the brew house as the nutrients for the yeast, of course. And the yeast looks upon these nutrients as its major food source for its reproduction. And in fact, if you think about this very simplistically, the primary aim of yeast in a fermentation is to reproduce. It's not to make beer. And I've said it on this slide, you cannot make beer without making yeast. And if you are able to do so, please tell me how you do that. I'm going to emphasize to you in this presentation that the availability of some key work nutrients is very, very important in governing not just the alcohol production properties of the yeast, but also aspects implicating flavor as well. Next one, Tobias. So Anne has very beautifully presented a similar slide indicating the yeast need for nutrients at different stages of its growth cycle during fermentation. But on this slide, I'm just going to emphasize to you one or two key points. And it was calculated many years ago that yeast actually produce alcohol during fermentation a lot faster when they are actively growing and budding than they do if they were non-growing or indeed stressed. And the other point is that growth is limited by lack of essential nutrients and ethanol toxicity, and we can regard high ethanol and indeed high sugar as a stress factor on yeast that can limit both growth and fermentative activity. So the bottom line on this slide is that attention to yeast nutrition is really key to ensuring smooth, consistent fermentations, and of course, avoiding stuck and sluggish fermentations. Next one, Tobias, thank you. So again, Anne has covered this very nicely. I won't dwell on this, but of course, we should look upon the wort has nutrients for the yeast to carry out fermentation and its growth. And remember, it's not just ethanol and CO2. We have a wonderful array of secondary flavor metabolites produced by the yeast, which will collectively play a very important role in dictating the beer characteristics, flavor, aroma, and indeed texture. The foaming properties of yeast are also influenced by key nutrients. And next one, Tobias. So one way of looking at yeast nutrition is to think what the yeast needs in order to grow its cellular biomass and its constituents. And on this slide, you can see the various percentages of protein, carbohydrates, lipids, etc. So we need to provide the yeast with the so-called building blocks for its cellular biosynthesis mechanisms. And I'll be talking a little bit about minerals and vitamins in this context shortly. Don't forget that yeast cells are heavily hydrated, containing about 75% water. So don't forget water in this conversation. And this is, of course, very important if you are doing very high gravity brewing, where there will be an osmotic stress on the yeast. Next slide. So Anne has covered this already, but just to recap, the principal nutrients in a malt wort are, of course, the fermentable sugars, the nitrogen sources, minerals, vitamins, oxygen I'll say something about, and I'll also mention a little bit about yeast foods shortly as well. Next one, Tobias. So the composition, the nutrient composition of your wort will influence many factors that are important for practical brewing. Firstly, the rate and degree of attenuation, the amount of yeast that's produced at the end of the fermentation, but also during fermentation, the flocculation behavior of the yeast will be influenced by key nutrients. And of course, as I've already emphasized, the final beer organoleptic characteristics will be impacted as well. And next one. So if we look firstly at the wort carbohydrates, this is just a typical profile of the main sugars that we generate in the brew house. And of course, maltose is the predominant fermentable sugar. And on this slide, you can see the dextrins. These are the non-fermentable oligosaccharides that normal brewing yeast will not convert into alcohol. In fact, if you make mistakes in the brew house, in the mash tun, for example, if you elevate the temperature too high, then you will correspondingly reduce the level of fermentable maltose and increase the level of unfermentable dextrins. And in fact, there was a very nice presentation. Some of you may have attended Andrew Patterson's Institute of Brewing webinar recently. Andrew's from Lallemand Brewing. And he was able to show that if you have a mashing regime, trying to recollect which key temperature, I think it was around 80 degrees Celsius, then that can be used to produce low alcohol beer, because you're reducing the level of fermentable sugars and increasing the level of unfermentable dextrins. Why you would want to produce low alcohol beer defeats me, but that's another question. The next slide, Toby, is what Anne has already covered, remembering that amino acids are the preferred nitrogen source for brewing yeast, and of course, as Anne has outlined, will produce a much more favorable flavor profile compared to if you were, for example, adding diammonium phosphate to the wort. Some small peptides, but not proteins, are not utilized. Brewing yeast is not actively proteolytic unless it's stressed. There is a rule of thumb that if your available nitrogen in the wort falls below around 150 parts per million, then you can have some problematic fermentations in terms of sluggish or even stuck fermentations. Okay, next slide. I'm going to move on to say a little bit about mineral nutrition. And on this slide, I've identified what are called bulk minerals: phosphorus, sulfur, potassium, and magnesium. And brewing yeast needs these minerals at relatively high concentrations, for example, hundreds of parts per million. Whereas the essential trace minerals, zinc, calcium, iron, etc., they are very often required at levels below 1 part per million. And then we have some toxic minerals, lead, cadmium, chromium, mercury, etc. And even if you have these toxic minerals at very, very low concentrations, you will kill your yeast stone dead. So please don't put a car battery in your fermenter, whatever you do. Next slide. So why do yeast need minerals? Well, for many cellular physiological functions, for growth, cell division, cell-cell interactions, that can mean flocculation, cell structure, membrane integrity leading to tolerant yeasts. But more importantly, for fermentation performance is the role of minerals in activating key enzymes responsible for the fermentative pathway. Next slide. Over the years, we've been interested in 3 important minerals in brewing: calcium, magnesium, and zinc. And I'll share with you some research to pinpoint how important these minerals are, mainly looking at their role in fermentation. But incidentally, the levels of these key minerals is not the same as the levels that you require to carry out an efficient and active propagation. But that's a different story. Next slide, thank you. So magnesium is a mineral very often overlooked by brewers, but it is absolutely essential for growth of the yeast and for fermentation. It's required for many, many enzymes of glycolysis, and it's also important in maintaining the structural integrity of yeast cell membranes. And in fact, magnesium can act as a stress protectant against high temperature, pH changes, and indeed toxic ethanol. Next one, Tobias. So magnesium and calcium. Yeast cells need to take in quite a lot of magnesium and at the same time spits out calcium, so that the intracellular ratio of magnesium to calcium in yeast cells is actually very, very high. The other complicating factor about this is that calcium has a well-known antagonistic role against essential magnesium-dependent reactions. In other words, if you have high calcium in the medium, this can suppress the active enzymes that are magnesium-dependent. And these are enzymes of glycolysis that will convert sugars ultimately to ethanol. Next slide. So I've tried to pinpoint this view here, that the yeast's view on the right-hand side is one where the yeast would like to take in a lot of magnesium and in fact spit out calcium, whereas in many cases the brewer's view is to have too much calcium in the work compared to magnesium. This is the wrong way round from the yeast's perspective. So the bottom line on this slide, I would recommend that you at least endeavor to maintain a high magnesium to calcium ratio in the fermentation medium in your work. Next slide. So we've looked at various fermentation media, from molasses to wine must to malt wort. In many cases, the magnesium to calcium ratio in fermentation media is not optimized and favorable for alcoholic fermentation. However, I've circled malt wort here, and fingers crossed, and hopefully your wort is of a favorable magnesium to calcium ratio, but there may well be occasions when you supplement with additional calcium salts in your brewing liquor, such as calcium sulfate. This is also called Burtonization. So what I'm trying to say is just be careful about the amount of calcium that's coming through to the wort prior to pitching your yeast. And as I've said, I would recommend maintaining a high magnesium to calcium ratio for consistent and efficient fermentation. Next slide. So yes, this is just emphasizing the word bioavailable. If you're measuring total minerals like magnesium, etc., that's interesting, but you should really be measuring what is going to be available for the yeast. And in fact, if you are able to increase the bioavailable magnesium in many fermentation media, you will actually find a benefit in terms of stimulating fermentative performance. This is not really fancy genetic engineering or rocket science. It's just addressing aspects of nutritional physiology for the yeast. Next slide. Okay, if magnesium is required at very high levels, hundreds of parts per million, then the next mineral, zinc, is still essential, but required at much, much lower levels, very often below 1 part per million. It's an essential trace element, however, and it's rapidly taken up by yeast during brewing fermentations. It's necessary for many enzymes, but a key enzyme activated by zinc is alcohol dehydrogenase, which is the terminal enzyme resulting in alcohol formation. Over the years, brewers have noticed that some stock fermentations may be due to zinc limitation or at least zinc deficiency. And if, for example, your levels of free zinc are below 0.1 part per million, then you may be in a problematic area. Next slide. So we've done work on looking at how quickly brewing yeast take up zinc from the wort at pitching. And on this graph, the pink symbols are showing you the levels of zinc in the wort at 0 time. In this case, about 0.5 parts per million. And you can see within the first hour or so, the zinc in the wort upon pitching yeast declines to almost 0. Where does that zinc go? It goes into the yeast cells. And in the dark blue symbols, you can see the zinc associated with the yeast cells. And I've expressed this as femtograms of zinc per cell. Of course, yeast is growing and budding during the fermentation, so that zinc will be diluted to the daughter cells when the cells divide and grow during fermentation. It means that at the end of the fermentation, the yeast is per cell, in terms of zinc per cell, is actually quite zinc deficient, and there is very little zinc left in the beer. Now, if you were to use these yeast for a repitching, for a next fermentation, and if that repitched wort was quite low in zinc, then you may have a problematic fermentation subsequently. Next slide. So you can actually visualize the zinc in the yeast cells using fluorescent dyes, and this slide showing you some flow cytometry of brewing yeast early in the fermentation. That's the upper graph, where you can see brightly stained yeast cells, whereas the bottom graph is showing you the yeast cells with very low zinc status later on in the fermentation. And next one, Tobias, thank you. This is a very busy slide, but it's really following brewing fermentation in which we've adjusted the initial wort zinc levels to different zinc concentrations. I'm not going through the graph in much detail, but I have summarized on the right-hand side that if you start with very low zinc, or indeed quite high zinc, then this can have a delaying effect on sugar utilization and fermentation performance. So what am I advising? I would advise that if you have your zinc levels in your wort on pitching between 0.4 and 1 part per million, then you should be in safe ground in terms of having a good rate of fermentation. Next slide. If you are suspecting zinc may be a problem in your fermentation, then of course you could supplement with some zinc salts, or there is actually a commercial brewing nutrient produced by Lallemand called Cervomyces, which is actually a zinc-enriched yeast nutrient. And on the right-hand side, we've taken some fluorescence microscopy of zinc-enriched yeasts, and you can see they're very brightly stained. And my student who took the micrographs called these galvanized yeast. Next slide. We've also looked at the consequences of zinc supplements on beer flavor, and I'm not going to dwell on this. There's a publication if you are interested, but if you do elevate levels of zinc beyond threshold levels, then there is an impact on flavor congeners, such as elevated higher alcohols, diacetyl, some esters, but lower acetaldehyde and unchanged DMS. Next slide. So in terms of the zinc story, zinc deficiency in wort leads to poor fermentations, inappropriate flocculation, less stress-resistant yeast. But if you have wort zinc levels at between 0.4 and 1 part per million, in my opinion, this would give you the best and most consistent fermentations. But remember what I said about zinc supplementations. You have to be careful on any subsequent effects on beer flavor. Next one. So to summarize some of the things we've found about magnesium and zinc, magnesium is essential for many of the enzymes of glycolysis that take glucose down to acetaldehyde. So magnesium, therefore, is required at very high levels. Zinc, however, has a very important role to play in the terminal enzyme called alcohol dehydrogenase. So insufficient zinc, insufficient levels of ethanol being produced. Next slide, thank you. So I'm leaving some summary points about minerals here. I won't dwell on these, but the top line on this slide is that you shouldn't really ignore minerals in wort because they are extremely important for dictating fermentation performance and indeed affecting the character of your beer. Next couple of slides on vitamins or vitamins. Next one, thank you, Toby. So vitamins for brewing yeasts are very important because vitamins are acting as helpers in metabolic processes, so-called coenzyme functions. Wort, if you've done your brewhouse operations correctly, should be generally sufficient in a whole array of vitamins, but there may occasionally be deficiencies in certain vitamins, such as biotin. And if you're at all worried about the levels of vitamins in your wort, you can supply with commercially available yeast foods. Next slide. This is just for your own reference, and it's levels of vitamins, typical levels of vitamins in malt wort. And you can see that the levels per 100 ml are generally in the microgram level. So very small amounts of vitamins are required, but they are nevertheless essential for many metabolic functions. Next slide. As I've mentioned, if you are worried about vitamin or indeed any other nutrient deficiency, there are commercially available yeast foods which can provide a whole variety of nutrients, such as increasing your yeast available nitrogen, extra sources of vitamins. Some yeast foods are also comprising sterols and fatty acids to make yeast membranes strong in the face of stress. And I've also given you an example of providing essential metal ions like zinc. And next slide. Very quickly, a mention about oxygen. Now, oxygen for brewing yeast is needed as a growth factor, not as a factor to stimulate respiratory metabolism. It's because of the Crabtree effect, and the high levels of sugar in wort means that yeast will predominantly ferment irrespective of the oxygen availability. So oxygen is required, a rule of thumb, 1 part per million of oxygen per degree Plato, but it's specifically required for membrane, sterol, and fatty acid synthesis. And there's also a role of oxygen needed in the lag phase to mobilize stored carbohydrates like glycogen. So in conclusion, I've listed here some key nutritional factors linked to poor yeast fermentation performance. Non-utilizable sugars, and I've mentioned if you have very high mash temperatures, then that can lead to high levels of oligosaccharides like dextrins, which are non-fermentable. Low levels of yeast available nitrogen, and Anne has given us some wonderful information regarding the influence of nitrogen nutrients in yeast physiology. Mineral problems, I've shared with you the effects of low zinc, excess calcium, insufficient magnesium. There may be problems with insufficient initial oxygenation and also occasional problems of vitamin deficiency. I haven't mentioned anything about chemical inhibitors or anti-nutrients. What would be an example there? I suppose if you left some caustic in the fermenter as you're pitching the wort, that would not be good news. And also stressed yeast. Generally, stressed yeast will have impaired nutrient uptake and poor fermentative performance. So next slide is the penultimate slide. And it's one I think you should remember, that yeast growth is coupled with efficient fermentation performance. So brewers need to find this balance. In other words, you don't want to have excessive yeast growth. You want to have a nice balanced fermentation. Yeast is wort food, and its composition is extremely important for good fermentation. So very fundamentally, poor yeast nutrition, poor beer. And then the final slide is just to thank you for your kind attention. My colleagues in the university in Abertay, I would like to thank Tobias and Didier for this invitation. And again, I will say cheers to Robbie Burns, and I hope you enjoy your morning coffee. Cheers. Thank you.
Thank you very much, Graeme. Again, very good presentation. And yeah, we have a lot of questions here, and I see if I can pull them up. And also, I think I should stop. No, that's closing the video. Yeah, don't know how to stop that. Perfect. So with the help of Natalie. So we have the first question. Do you recommend adding nutrients during yeast propagation? If so, do the addition rates differ significantly from those used in fermentation?
Yes, good question. Sorry,
yeah.
Do you want me to try and answer, Tobias?
Yeah, absolutely, yes.
Yes, in fact, I know several brewers that actually do exactly that. They do add additional nutrients at the propagation stage, and I think in some cases it might be a good place to add additional zinc if you were worried about zinc deficiency. So yes, that's a good question, and it's going to be a smaller vessel compared to the large-scale brewing fermenter as well. So practically speaking, I think it's an easier operation.
Mm-hmm. And I guess in wine yeast, there's not a lot of propagation, so leave that. Next one is, what is the relationship between DMS formation and nitrogen depletion? Is this also yeast dependent? Thank you. And this is from Raphael.
Well, yes, DMS comes from grapes, for from the medium, for sure. Mm-hmm. And the yeast can impact some. Depending on the yeast, you can preserve the potential of DMS, dimethyl sulfur, or at the contrary, you can decrease this PDMS. So there is the genetic background of the yeast, because some genes have been already identified for contributing to this preservation or not of the DMS. And we have done a few years ago some work looking at the impact of a specific nutrition during alcoholic fermentation in case of nitrogen depletion. And we have seen again that organic nutrition had a better impact in the preservation of the potential of DMS, which is something that I don't know for brewers, but some people are looking for, because can be responsible. The contribution at the aromatic point of view is nice. Contrary to ammonium. So that's what I can say. No more, because it's quite a new investigations on such topic.
And could I follow up a question to you about the pantothenate deficiency leading to higher H2S? Mm-hmm. Is that mainly in wine yeast, or is it also a situation in brewing yeast?
I guess it can be in brewing yeast. It's because the pantothenic acid is the substrate of the precursor, of acetyl-CoA. And without enough acetyl-CoA availability, its acetyl-CoA is directly included in the assimilation of the homoserine in the sulfur, the pathway of the yeast. So I guess at least for Saccharomyces cerevisiae, if it's a Saccharomyces cerevisiae, it works the same. It works really the same.
Mm-hmm. Thank you.
Yeah, and I can confirm we have that in breweries, and often when we get questions or complaints that there's too much sulfur flavors, often the addition of a little bit of calcium pantothenate helps and solves that problem. Good. This is a question for Graeme, I guess. How high is the calcium-magnesium ratio, or what is the recommended calcium-magnesium ratio? I guess in terms for the yeast, not.
Yes. If you look inside a yeast cell, the ratio of magnesium to calcium is 1,000 to 1. Now, you're not going to achieve that in work, but at least, as I tried to emphasize in my presentation, at least if you try to have a high magnesium to calcium ratio. Now, for example, if you have worked with, say, 150 parts per million magnesium and 50 parts per million calcium, you're in the right ballpark. But actually, the yeast would like to see that ratio even higher. But I'm talking in practical terms, because wort is wort. You have calcium, you have magnesium. So you're never going to get 1,000 to 1 in your wort. But at least if you have it, I would suggest 2 or 3 times the level of magnesium compared to calcium, then the yeast would be happier.
Thank you. The next question, can cysteine and methionine, sulfur-containing amino acids in excess lead to sulfur notes in beer? I guess the same in wine. And
well, usually it's rare to have free amino acids in excess at the end of the fermentation. So usually the yeast will uptake all these free amino acids, like all nitrogen sources in the first part of the fermentation. What can happen in case of nitrogen depletion at a certain point during the stationary phase, for instance, of the fermentation, the yeast has a possibility to catabolize and to use its pool of intracellular amino acids, such as cysteine and methionine. And by using the carbon skeleton, I don't know the word, the carbon part of such amino acids will release the sulfur part of this sulfur contains amino acids. So this can be risky at the end of the fermentation, but it's if we avoid from the beginning a nitrogen depletion, we shouldn't have such kind of phenomenon.
Thank you. Graeme, you want to add anything?
Oh, no, that's fine. Thank you.
That's fine. Perfect. Good. Thank you very much. Next one. Hi. So there are nitrogenated volatile compounds, thiolated compounds. If so, would this imply that nitrogen nutrition is as important as sulfur nutrition for the release of these aromatic compounds? And I guess that's for you again.
I'm not sure if I understand the question. I don't see the question, but if there is a link between
nitrogen and sulfur. So usually, if you have, I guess these precursor, he means, yeah.
Positive, you mean positive sulfur compounds like the volatile thiols?
Yes, I guess.
Yes. So yes, for sure, because it's not only nitrogen, but it's as well vitamins and minerals, which are key, because as Graeme has mentioned, they are directly linked, and they are enzymatic cofactor, really important, and they increase the expression of some specific genes as well. And the beta liase is also very the expression of such kind of enzymes has been demonstrated to be linked with the level of minerals, vitamins, nitrogen, all these such kind of nutrients. So yes, there is a direct link with specific yeast nutrition and the capacity of the strain to convert, to uptake, to convert, and to release the volatile thiols.
Mm-hmm. So it's
if I understood the question.
Yeah, no, I think this is was a question. So not just the precursors from the media, but also it's very important to have the proper nitrogen source in order to for the yeast to take up, and then basically
and the enzyme stimulation and the
enzyme stimulate. Yeah. Mm-hmm. Okay, perfect. That's one for Graeme again. What level of calcium in wort is too high?
Actually, we have done some work in synthetic wort with 0 calcium. 0. Okay, it was at the limit of analytical detection. You're not going to believe this, but the yeast was perfectly happy. It didn't flocculate because there is a role for calcium in flocculation. But seriously speaking, yeast does not like to see a lot of free calcium for intracellular metabolic functions. There are some key enzymes, of course, that are calcium dependent, but if you compare that with the number of enzymes that require magnesium, then there is no competition.
Mm-hmm.
Any enzyme that transfers phosphate is a magnesium requiring enzyme. You cannot make ATP without magnesium because the substrate is magnesium ADP, just for example. Mm-hmm. Sorry, I'm digressing. What was the question?
The question was actually, what is what level of calcium is too high? So is there actually a certain number, like for zinc concentrations, for instance, which becomes toxic?
Well, what did I say before? If you have 150 parts per million magnesium and 50 parts per million calcium, if it was the other way around, 50 parts per million magnesium, 150 parts per million calcium, that's the wrong way around from the yeast's perspective. So I would suggest if you are above 100 parts per million calcium, then there may be some problems in terms of the consistency and efficiency of fermentation.
Mm-hmm. Okay. Good.
I should say,
yep,
sorry, Tobias. What I'm talking about is the levels of these minerals that you measure in clear wort before you pitch the yeast. Don't measure it in the wort running out of your mash tun, because you're going to, in the boiling process in brewing, you're going to be precipitating a lot of calcium in the trub. Do you get my point?
Yep.
So any analysis of minerals should be done on the clear wort just prior to pitching your yeast.
Perfect, thank you. Next question. Can you make recommendations regarding additions to yeast in storage, cropped post-fermentation, stored at 2 to 5 degrees Celsius in fermented beer? For instance, unsaturated fatty acids, zinc, sterols, salts, or even out osmotic pressure, etc. Is there anything they could do to,
Yeah, that's an excellent question. And I'm not giving any secrets away here, but I know of some brewers who oxygenate yeast in storage tanks. If you think about that for a second, it makes a lot of sense, because the yeast needs oxygen to re-energize its membranes, its sterol biosynthesis, its oleic acid biosynthesis. So if you're able to provide the oxygen at that point, then you might not need to add or aerate or oxygenate your wort prior to pitching. So, okay, I know the question was about nutrients in yeast storage, but oxygen you could regard as a nutrient in that sense. And I think from a yeast physiology point of view, that is really quite an intelligent approach.
Mm-hmm. And I guess also addition of zinc, if you really have a deficient wort, would be good, because you don't get a lot of the chelation that you have when you add it actually into wort, when it's just the yeast itself in water, for instance. So,
and as I indicated, the yeast will take up the zinc if you add it very rapidly as well. So that would be another example. Yeah.
Mm-hmm. Good. Thank you. Next question. Great presentation. Thanks. Question is, nutrient content in wort is well recorded as very important for good performance, and it is also possible to help some high ABV alcoholic fermentations, adding some commercial nutrients after fermentation started. That was a long sentence. So, in your opinion, what is the moment during attenuation from which it is not recommended to add further nutrients in order to do not impact on final flavor profile? I guess, Anne, that's something you have a lot of work done on addition, or when to add.
Yes, we advise at the beginning or maximum 1/3 or the mid fermentation, but my reference are not exactly the same as yours. So, but it's important before the stationary phase, I would say. It's and finally, especially at the end of the exponential phase, because at that point the yeast has already consumed all the nitrogen present in the must, in the wort, or in the medium, and so it's at that point it's key to add something in order to continue to uptake the sugar or whatever, which kind of sugar, and to continue the fermentation, to avoid any stuck fermentation or sluggish, which is even worse, and which can lead to flavor, VA, sulfur compounds production, etc. So beginning or 1/3 or at the end of the growth phase is really the optimized moment. If we want to drive some specific profile, aroma profile, for instance, we know that if we want to favor the uptake of precursors to release at maximum volatile thiols, for instance, in case of you have a good amount of precursors, the best moment is at the beginning, because the beta-lyase activity of the yeast is really very active during the growth phase, the multiplication phase. So at that point, we will really stimulate all the enzymatic system from the beginning. If the target is aroma.
Graeme, do you want to add anything?
No, that's fine. Yeah,
that's fine. Good. Next question is, I found that reusing yeast sometimes change flocculation capability. What could be the reason for that? Graeme, do you have any?
How long do we have?
Yeah, flocculation is still
yeah.
A lot of unanswered questions as well.
You could write a book on the factors that can influence flocculation, but at the top of that list would be the genotype or the strain of yeast that you're starting with, and the expression of the classic flow genes. But in terms of worked nutrition, I've mentioned calcium already. There is a well-defined role for calcium in flocculation, but there are also roles for other nutrients in dictating cell-cell interactions during a fermentation. Temperature, agitation, a whole range of other things. Tobias, I'm sure you could add to that list as well.
Yeah, I mean, as I said, this is as you said, it's a huge topic, and there's still a lot of open questions. We've seen that sometimes zinc addition or zinc depletion can lead to less flocculation, but then on the other hand, we also seen that too much zinc has the opposite effect. So it's always finding the right balance, but looking at zinc or minerals could be one thing, calcium, things like this. Yeah, unfortunately, we don't have the absolute answer for that at the moment, I would imagine. Or Anne, do you have anything in wine in terms of flocculation?
No, exactly what Graeme said. I don't have anything.
So that's
to add. Yeah.
Perfect. Next question is, were these media made with demineralized water? I believe that refers to your slide, Graeme, where you showed calcium, magnesium ratios in different media.
Yeah, good question. The work we've done in our own laboratory is with Scottish East Coast water, which is quite soft, to be honest. But we can adjust in experiments to look at the levels of these minerals. The work we've done in molasses relates to rum and biofuel production. And here the levels of calcium are problematic, especially if you're doing a distillation, because you can have scaling in the stills and downstream processing problems. So a lot of alcohol producers that use molasses now tend to acidify molasses with sulfuric acid. That will bring down calcium. And actually that artificially or in a way lowers, raises the magnesium to calcium ratio if you precipitate the calcium. And that actually benefits fermentation and also benefits downstream processing as well.
Thank you. Next question is, what can you do to increase zinc within the Reinheitsgebot? No salts may be added. Yeah, that's a tricky question. It's very difficult, actually. So in Germany, we're not allowed to add anything to the fermentation. So at one point we thought the zinc-enriched yeast actually would be, but I believe the German authorities are against that, so can't claim that anymore. So yeah, very difficult to say what you could actually do.
You could galvanize your fermenters.
You could galvanize your fermenters. I know that some breweries have zinc plates as part of their equipment, which disappears over time. That could be an addition rate. But yeah, so I don't have an answer there. It's also a lot, the malt itself provides a lot of zinc, so but a lot of that is actually lost in the spent grain, and it's also chelated with the phytic acid. So if you could avoid this chelation process and have more ionic zinc available somehow, that could help, but I don't have an answer to do how to do that, actually.
There may also be a zinc contribution from hops as well, Tobias.
Yes, sorry, yes, which comes later, and but I think one of the key problems is, and that's what we had with the bioavailability, is that a lot of the zinc is actually chelated into two large molecules and then can't enter the zinc. So even if you do an AAS analysis and you have 0.3 ppm zinc, this might not be all available to the yeast itself then. So, and there was a great paper by a Hungarian group that showed actually free zinc and bound zinc from 10 different industrial breweries that they analyzed, and was quite interesting. So I'm sorry, yeah, we don't have a real answer on that. Now the next question is the same question we had already with the demineralized water. I guess I go to the next one. Is the new Saccharomyces vacuum packaged product from Lallemand different from the unvacuum packaged one you showed in the slide? Can Saccharomyces lose its nutritional value based on time and storage conditions? What is the best way to store Saccharomyces when unopened, and once opened? Can zinc concentration in wort be calculated based on weight in grams of Saccharomyces per hectoliter of wort for, no, I can't read that. I need to pull that down, sorry. For example, 1 gram of Saccharomyces added adds 1 ppm of zinc in 100 hectoliters. Graeme, you want to answer? I mean, can I answer a couple of questions, a couple of these topics? So the difference between the Saccharomyces vacuum packaged and the Saccharomyces in the loose packs is that one was the loose pack was inactivated yeast, whereas the Saccharomyces in the vacuum packs is actually still a living yeast cell, and that has to do with part of the regulation question. So if you have a living cell, it's part of your ingredient, whereas if you have a dead yeast, it's a yeast nutrient or an addition, a yeast additive. So that's the main difference. So one is alive, the other one is dead. And that's why we also recommend to add the yeast either to the boiling kettle 10 minutes before the end of the boil, or if you don't want to do that, you could also measure the amount of Saccharomyces that you want to add, and then put the resuspend or spend that in water and autoclave it, and then add it directly to the fermenter. So if you want to avoid losses, potential losses in the whirlpool. Saccharomyces is stable over time. It's really the yeast. The viability of the yeast is not important in that term. It's not supposed to ferment, and as I said, you should kill it actually before you add it. So and the zinc doesn't disintegrate or disappear, so the zinc concentration will be the same. So from that point of view, there's no deterioration over time. What you should do in terms of storage, it should get kept dry, otherwise it tends to clump, and then it will be difficult to actually measure. It doesn't need to be stored in a cold place, because as I said, the viability is not really an issue here. So even if the yeast becomes dead, the product is still functional. Then in terms of adding zinc, so we have a certain range of zinc that is attached to the cell. That usually the specification is between 50,000 to 60,000 ppm. So you can calculate 1 gram how much zinc you would add. If you add it to the brewing kettle and then it goes through the whirlpool, you will lose some of the zinc because the yeast cell itself is it's not solid and will be some of that will go out with a drop. And we did experiments. So if you add 1 gram per 1 gram per hectoliter, I think that's right, 1 gram per hectoliter. And so usually, in theory, you would get 0.5 ppm zinc, and we found in the end in the fermenter 0.3 ppm zinc. So there is a little bit loss, but this is already calculated into the addition rate that we recommend. If you want to use 100% what you add, I would recommend to, as I mentioned before, suspend the yeast, autoclave it, and then add it directly to the fermenter, and then you have the full value of the zinc. Graeme, do you want to add anything to that?
No, that's fine, Toby. Thank you.
Okay. Anne, I guess no. Good. What will be the most important nutrients to consider to build resilient yeast that lasts? Now it moved. Oops, that question is gone. No, here. What would be the most important nutrients to consider to build resilient yeast that lasts longer in storage between pitching? Graeme, do you have any?
Certainly do not discount the role of oxygen, because there is a very specific role for oxygen in making strong membranes, which are going to make yeast resilient in the biosynthesis of ergosterol and oleic acid that collectively will make the membranes more stress resistant. So don't forget oxygen, but we've also done work with minerals as well, and the role of magnesium and zinc are also playing a role in stress resistance of yeast as well.
Mm-hmm. With the addition of sterols itself to the yeast?
Yes, some commercial yeast foods are based on sterols and unsaturated fatty acids, and under the right conditions, the yeast will take up preformed ergosterol and oleic acid to incorporate into their membranes. So yes, that is another approach.
Yeah, I was thinking about GoFerm that is used a lot or developed actually for the wine fermentation, and this is used as a yeast rehydration nutrient. But have you used it also in fermentation or adding to liquid yeast? Anne?
No.
No, okay.
No, because it's really important to use this concentrated inactivate yeast autolysate with this high amount of sterol. It's very important to use the right concentration between the yeast and the right ratio between the yeast and this yeast autolysate with high sterols, in order to be sure to have this micellar formation and the integration, the solubilization of the ergosterol and the sterols, to be able to be internalized in the yeast cell membrane during the rehydration step. So no, it has been, well, we have done some tests. For sure it can be efficient, but the efficiency is decreased compared to the use in rehydration.
Okay.
Good. Tobias, could I also add that I know of some brewers who add a little bit of trub sometimes back to the fermenter. And the idea being that there are some growth factors, maybe some membrane factors that are going from the trub back into the fermenter to help the yeast. Distillers do this. It's called backsetting. They add stillage back to the fermenter, which is providing additional nutrients as well.
Yeah, I heard that as well, that brewers use that. But you have to be careful because it also can happen then sometime if you use too much on your flavor. I've heard that dry yeast come with more nutrients than liquid yeast. Is that true? And you have any? Or Graeme, sorry.
Well, if you use dried yeast, remember it has been pre-propagated in an aerobic respiratory system. So you might, well, the yeast is going to have what used to be called an oxygen credit, which means that they have very nicely preformed membranes with enriched sterols and fatty acids. So I'm going to pass this over to Tobias, because some brewers say they don't need to aerate their wort if they're using dried yeast because of this oxygen credit, but maybe you could comment.
Yes, that's true. I mean, we did a lot of experiments where we did fermentations with and without aeration, and you don't really need to aerate dry yeast because, as you mentioned already, in the process we aerating heavily, and we have a high concentration of sterols and unsaturated fatty acids in the yeast. So aeration in a regular wort is not really necessary. But this only works if you use a dry yeast per se. So if you harvest the yeast and basically have liquid yeast and then repitch it, that whole benefit is gone if you want. So this only works if you use the dry yeast itself. And then, I don't know, in wine, do you aerate a lot?
No. Yes, we can, during a fermentation, you mean?
Yes, start of fermentation, basically.
Yeah, we can aerate, but we, again, during the first stage of the fermentation, it can be tricky because there is lots of oxidase, polyphenol oxidase, for instance. We can consume very quickly the oxygen instead of the yeast. So it's better to wait a little bit to have a sufficient multiplication before to aerate, to be sure that the yeast will benefit really of this oxygen addition to synthesize their own sterols and saturated fatty acids, etc. And just now coming back to the question between liquid and dry, active dry yeast, for sure, I'm convinced that active dry yeast has been prepared to have a strong yeast with all the package in terms of specific nutrients, and to be able to face the hard life, which is alcoholic fermentation, which is very hostile for the yeast. And so I guess the active dry yeast are much more prepared to ferment compared to liquid.
Well, and I can confirm that just looking at minerals, for instance, when we compare or we analyze liquid yeast that is in the market, and then dry yeast, zinc, for instance, or other minerals are much higher because we're adding quite a huge amount to them.
For sure.
We're running out a little bit of time. I guess we can do 2-3 more. So in my groundwater, I have 90 ppm of calcium and 30 ppm of magnesium. Don't know how much the malt contributes. Should I add magnesium? Is there an upper limit to magnesium? Do I have to product of FAN and zinc? Do you have a product of FAN and zinc? Sorry. Yeah.
So the first question about the minerals in water. Yeah, of course, the minerals in your starting brewing water are going to be important, but I've already made the point that if you are wanting to know the availability of minerals for fermentation, you should be measuring in the clear wort after it's gone, after your brew house and wort boiling and you've produced your clear, cool wort for fermentation. That's when to measure. So, for example, if you're starting with groundwater that is higher in calcium, it may not be reflected in the wort calcium because there will be precipitation occurring in the brewing process. Do you get my point?
Yeah, no. Well, I get it at least. And and for the question, do you have a product of FAN and zinc? Yes, we do. We have a product out that is called Yeast Life that contains both zinc and nitrogen. And the other products, either the Saccharomyces is just zinc, and then we have Fermate K, which is a lot of nitrogen, but unfortunately no zinc. So this would be the best product if you want both in one product. Have you done any work on nutrient requirements in quake yeast? I've read that some research Escarpment Labs did on this regarding elevated nutrient requirements, FAN consumption 180 ppm, but wondered if you have any view on this. Graeme? No, I have to say we haven't looked at that yet either. And I guess in wine, quake yeasts are not, well, are they used in wine?
I don't understand this.
Quake is Quake is a new yeast that comes from Norway. It's very temperature.
Okay.
Quake. Yeah, depending on how you pronounce.
Sorry for the. No, it's me. I don't know how to pronounce. Okay, quake. Well, yeah. No, actually, it's well. Thanks to Didier, we will have a look on such yeast, but
it's a unique yeast with the temperature tolerance and the flavor profile it produces at high temperatures. But
okay, no, not yet. It's it will be in the next weeks.
It's coming. Good. Thank you. Brew kettle metal effects: aluminum versus kitchen grade stainless steel. Wow. I have to say I don't have any answer on that, Graeme.
He means aluminium, not aluminum.
Yes.
Actually, aluminium is quite toxic to yeast, so I would not recommend any brewing vessels with high levels of aluminium, to be quite honest. Actually, we've done some work in Brazil relating to molasses, the sugarcane molasses process. Sometimes the aluminium levels are quite high in the molasses for fermentation. But if you elevate the magnesium levels, this can mitigate the effects of aluminium toxicity. That's just another story, but aluminium is not is quite toxic.
Yep. Okay, thank you. Does the age of grains make a difference in the amount of nutrients available? Which nutrients suffer most in old grains? I guess that we should ask maltsters. Or Graeme, do you have any?
In old grains, I would be more worried about mycotoxins than nutrients, to be quite honest, especially if the old grain has been stored in high humidity, high temperature. There's always the risk of fusarium contamination.
Next question. What do you think are the basic gears, tools for an acceptable yeast nutrition, like O2 tanks, Erlenmeyer, etc.? Greetings from Ecuador. I'm not sure I understand that question completely. So what are the basic gears and tools? Or what would you recommend to add, I guess? Oxygen versus air?
Yeah, maybe have good analytical chemistry as well to, determine your baseline of your standard wort. Not just, for minerals and vitamins, but, the levels of yeast available nitrogen and fermentable sugars. I think you need to have some good chemistry to start with, I think.
So you need basically to know what you have in your wort in order to supplement them. Good. I think these were the last. We do two more, and that's really it then. Given a normal wort, would you recommend magnesium supplementation, or would this solely be based around maintaining the calcium-magnesium ratio? So would be dependent on the calcium level. So would you recommend adding magnesium anyhow?
Again, going back to my previous answer about analytical chemistry, I think you should at least know your baseline minerals before you even consider any supplementation. having said that, in general, some small magnesium supplementations are a benefit for fermentation progress. But remember, we're making a flavorsome product. We have to think about flavor effects of any supplementation, whether it's minerals or anything else.
Mm-hmm.
You know, this is your bottom line. It's a flavorsome product we're talking about.
Yeah, perfect. And I would say this is the last one. Sorry if this was said, but does Saccharomyces supply copper as well? As one of my previous questions, and if Saccharomyces provides copper, can we calculate copper concentration added to wort based on weight of Saccharomyces? Saccharomyces does not contain any copper. So I guess that's the answer to it. So we don't calculate also the addition rate based on weight. So I would stop that here. I know see we have still more questions, but we went over time already quite a bit, 15 minutes. So I thank you both, Anne and Graeme, for fantastic presentation. That was really interesting. I learned a lot as well. And I thank everybody who listened and stayed put for such a long time, almost 2 hours. And stay tuned for more webinars to come. So thank you very much. Thank you both, Graeme and Anne, and see you next time.
Thank you, Tobias. Thank you, Nathalie. And thank you, Didier, for the organization.
Merci beaucoup, Anne.